Now this transformation is used in molecular biology to purposely engineer a cell today. We do this in biotech over in Newark. We will force cells to uptake DNA and they become different. So you better watch who you hang out with. Okay? So Griffith realized there's a particle here. This Mendel particle was transferred from these, maybe these S cells, when they're dead, they're leaking molecules, as they are. And these R cells have to acquire some of those leaking molecules, and they became stably transformed such that their offspring exhibit the same trait, their new trait. That is, all the descendants are S cells. Those recovered and their descendants beyond and beyond. So scientists realized, well, heck, we don't know what the molecule of a gene is. We don't know if it's DNA or protein. Well, if we can recreate this system in our laboratory and isolate these particles, we know what it is, right? And there was a team of American scientists that did this for 10 freaking years, because chemical methods were rather crude in the 1930s. This is Avery MacLeod and McCarty. Okay, they did this work in the early 30s, late 30s and early 40s, published in 1944, right near the end of World War II, and they did a very extensive work. Okay, they took these S strain cells. They had this particle that could transform an R cell into an S strain, and they broke it into all these components of the cells. They separated out the S strain cells, RNA and protein and DNA and lipids and carbohydrates, because they wanted to know which of these is the gene that could transform an R cell to an S cell. Okay? So they isolate these particles, then they tested for transformation of the R strain cells to see which would transform the cells, and only DNA would transform them. Only DNA would work. So they thought, okay, that's it. DNA is the particle of information. It's the molecule of inheritance. They were right, but most scientists said, yeah, I'm not so sure. Your methods are so crude. How do you know this is 100% pure DNA, not a little bit of protein? And they couldn't say that it was pure. The methods of the day, they could not say conclusively that their extracts were 100% pure. It could be that if there's a 0.1% contamination, that could have been enough to transform them. So they couldn't rule out contamination of their extracts, but they were right. So most scientists were still a little skeptical that DNA was a genetic molecule because they still couldn't understand how such a simple molecule could carry information. Proteins are complex. It would make sense that a complex molecule would be the molecule of inheritance, not this simple DNA with only 4 monomers in its polymer. So it required more confirmation. And this involved an effort by a couple in the early 50s, Alfred Hershey and Martha Chase. They did the famous Hershey-Chase experiment. Now Griffith, he just kind of stumbled on this transformation. He was really looking at what caused certain strains of bacteria to be pathogenic. And he observed something and then documented it carefully. Avery, MacLeod, and McCarty, they tried to identify the transforming particle, and they got it right, but they couldn't confirm that their extracts were contaminant-free. Hershey and Chase, their goal is to decide conclusively, is DNA the genetic molecule of inheritance, or is it proteins? They need a simple system where the conclusion is clear and without conflicting possibilities. So what they decided to do was study infection of a bacteria by a virus called a bacteriophage. Now, every living organism, every living thing on the planet is subject to viral infection. And a virus is really a parasitic gene encapsulated in some sort of protein compartment. Okay? And viruses are not considered to be alive because there's one thing they cannot do. They cannot reproduce themselves. That's why they're not considered to be alive. You can take a virus and break it into its component parts and store it in a laboratory jar on the cabinet like any chemical, mix it back together, and they become infectious. So that's pretty spooky. Okay? You can't do that with a living cell. You can't take all the chemicals of a cell in laboratory bottles and mix them up. You got a big mess in your hands. But you can take the chemical components of a virus, mix them, and you've got active viruses. So they're on that cusp between molecules and living things. But they're not considered to be truly living because they can't reproduce themselves. How do they reproduce themselves? They have to infect a host cell and inject their viral genetic molecules into the host cell and let the host cell copy their genetic molecule and then use it to make viral proteins, and they can escape the cell and infect more target organisms. So the host cell that the virus infects becomes the viral nursery. The virus sort of commandeers the host cell to reproduce itself at the expense of the host cell. Okay? Now bacteriophages are ideal for this because they are the simplest viruses known. They only have two components. A protein compartment called a capsid, okay, that's coded for by the viral genetic material. So this is the protein component of a bacteriophage. Sometimes they're just called phages. Okay? I'll write that word down in case you don't know that. Bacteriophage is a type of virus that infects only bacteria. Okay? So the capsid is the protein compartment, and inside the capsid is the genetic molecule of the virus. In this case, it's DNA. Genetic molecule of phages is always DNA. Now there are lots of viruses that use RNA as a genetic molecule, like HIV is an RNA virus. Okay, but all phages have DNA. So again, a virus can only reproduce itself by injecting its genetic molecule into the host cell, in this case the bacteria. So they could track what was injected. Was it the capsid injected or the DNA? They would know for certain which is the gene, right? But how do you track that? That's the challenging part. Well, they're going to use radioisotopes. Okay? It turns out that proteins, they look for some element that didn't overlap between DNA and proteins. And for proteins, what's present in amino acids but not in DNA nucleotides is sulfur. That's unique to proteins. There are some amino acids that contain sulfur only. For DNA, it's phosphorus. There's no phosphorus in any amino acids, but it's part of the sugar phosphate backbone of DNA that we'll see in a bit. What you're going to do is you're going to use a radioactive isotope of sulfur, in this case sulfur 35. I think it emits beta particles, as does the radioactive phosphorus P32. So you're going to establish two separate batches of these phages. In one batch, you label the protein capsids, and the red here indicates the radioactive label. So the capsid is radioactive. You can detect the Geiger counter or scintillation counter because it emits particles. In a separate batch of viruses you're growing, of phages, you've got the only radioactive isotope is P32. So the DNA is labeled, not the capsid. Okay? You allow these to infect the host cell. Of course, this is what we know happens now, that the phage, I mean, these phages look like this actually. Electron micrographs show they look like these lunar, this is not the artist's sort of version. This is an actual, I've got photographs I could show you, but I won't today, they look like lunar landing pods. They attach and they inject like a syringe their genetic molecule into the cell. Of course, we know it now to be DNA. Okay, you allow the phages to infect the host cells, this time in case it's E. coli. Okay, you then take, after you allow for sufficient time for infection, you take this mixture of virus and bacteria, you put in a blender so you can knock off the attached, whatever on the outside of the phage, whatever on the outside of the bacteria that's not injected, off, and it floats in the supernatant. You then spin the cells to the bottom of your, you take this liquid out of the blender and you spin it in a centrifuge, and of course heavy things pellet to the bottom, okay, in your centrifuge tube. Okay, after centrifugation, dense things at the bottom, so you got a pellet here, which is the cells, the bacteria, and floating up here is whatever you knocked off in the blender. You take your Geiger counter, you find out where's the radioactivity. And when they, in the separate batch, when they had the proteins labeled, the radioactivity was up here. It was not in the cells, so protein was not inside the cell. When they had a separate batch with the DNA labeled, they again allowed for infection, then blended off the outside attached capsids, spin it in the centrifuge to pelt the cells to the bottom, okay? Because now the radioactive DNA is inside, the radioactivity was here. So they knew conclusively that the DNA was injected in the host cell, the DNA is the molecule of the gene. Now everyone says, you got it right. Now we know DNA is the molecule of inheritance, but we don't understand how it works. How can it carry molecular code? How can it be copied from generation to the next? And this is 1950, and so what's about to happen is a race for fame and fortune. There's the Nobel medal, because now the leading molecular biologists in the country, the biochemists, they knew If they could figure out the structure of DNA such that they could explain how it carries a molecular code and can be replicated faithfully from one generation to the next, they would win the Nobel Prize, which was true. Who are the participants in this race for glory? Well, here was the leading biochemist in the 1950s. He was at Caltech in Pasadena. This is Dr. Linus Pauling. He already had one Nobel Prize for describing the nature of the chemical bond. And he had just completed a description of one of the first using X-ray crystallography to determine the 3D structure of an important protein called collagen. So he was an expert at determining three-dimensional structures of macromolecules. If you were going to bet, this is the guy you bet on. Okay? His competition, it turned out to be, was a team in England. The team involved Dr. Maurice Wilkins, who was trying to precipitate DNA into crystals so they could do X-ray crystallographic photographs. And they recruited back to England an English girl who was working in France, Dr. Rosalind Franklin, who was brought back to France to help prepare the DNA samples and do the X-ray work so they could take pictures and see if they could deduce the structure of DNA from these X-ray photographs. They were at King's College in London. Meanwhile, up in Cambridge were two drinking buddies. This is Dr. Francis Crick, who was pushing 30 years old. He was one of those guys, when I was in grad school, there's always a few guys that are there for like a decade and a half because they like grad school. They're not trying to get out. Why would I? There's a real word out there I don't like. So he was in the Cavendish lab at Cambridge, which is one of the leading X-ray crystallography labs in the world, looking at protein structure using X-ray crystallography. We'll talk about how that works. This is his new drinking buddy from Indiana University. He come to England to do a postdoc in the Cavendish lab to learn to do X-rays. He is a new PhD from Indiana Bloomington and was known to be a disaster in the lab. They became drinking friends and decided